Very good morning to all of you. Thank you very much for joining in. Today is a very important day. We are going to talk on the welcome to this webinar on analytical instrument touching your life. Now, this is a interesting concept. Why I chose this topic is we have to be concerned about all about around us, the environment, and what is around our environment, what are our requirements, etc., and how analytical instruments are helping us to make sure that we have a very good life. And it will be surprising, and also it is very important for our students to understand how important chemistry and analytical instruments are in our life. So with these thoughts, I have really coined this talk, and it will be segments. A few things is to say what are our requirements. Number two, how these instruments are going to help us in our daily life, and then what are some of the principle under which it works, the chromatography and spectroscopy works, and then we'll also going to see what is around us in the nature, what are the things we have learned from the nature, how we are nurturing the nature for our own benefits. So these are the few aspects that I want to touch. I hope that I will be able to do this in 1 hour. What are our basic needs? Our basic needs are very simple. One is we want a very, very cleaner atmosphere, a cleaner environment to live in. Cleaner is to do with all the environment around us, whether it is air, water, earth, whatever. And then healthier. We want to make sure that our food system, our pharmaceutical, our drugs that we consume, whatever to do with our health, we are concerned about it. Safe. We want to safer. As we step outside, we want to make sure that we have safety. Even when we are in our houses, we want to have safety. Now, the safety comes from the materials that they use in the building of the construction. And when we go out, it depends upon whether people are driving safely or whether there is drunken driving, or then we want to see whether there are any hartals, any problems, whether any shooting, or whether there is any, arson, looting, explosives. All these things are coming into the safer, including also we want to make sure our children are going to school where it is safe, the environment is safe, there is no drug peddling, etc., and our children are safe. So we want to make sure in every way that we see that the environment that we live in is very safe. That is what we need. However much money we may have or we may not have, but what is important is these are the aspects that we need. So the title analytical instrument touching your life, and that is they add quality to your life from the time that you get up till your time that you go to bed. And right when you open the tap water, you are worried, is this water safe? You want to look at the color of the water, you want to see whether there is a turbidity, whether there is any materials floating around, and then you want to see whether there is any smell. You want to make sure that the tap water is safe before you can put it into your water, into your mouth. And when you right now when you use your toothpaste or any of these materials, any cosmetic materials, you are worried whether they are safe to be used. And whether it is a hair cream, soap, shampoo, anything like that, and perfume, you are always worried whether this is of good quality, will it have any problem for me? And continuing, when you wear your colorful dress, whether the colors are safe, whether they are causing cancer. There was a time when some of the dye stuff which was used on the clothes, they used to really cause the skin cancer. And then when you wear your footwear, there are a lot of things, there are a lot of additives which are added into wear, a lot of materials are used into the leather tanning. So all those things can cause problems for you. And when you take your breakfast on your breakfast table, how safe is the food? What are the colors that is being used? What are the additives they have in the milk, etc.? So everything is very worrisome. And when you take your morning medicine, for example, you want to make sure that the medicine is good quality, good companies make it, they go through a good process. And when you go to a car, you are concerned what type of petrol you are using, what type of oil you are using. All this has to do with the chemistry and instrumentation. Till you take the last pill of the day, if at all you are taking anything, or if you are taking a glass of milk also, you are worried whether is it safe. So we want to make sure that your day after day is safe. And this safety is assured to you because there are people working round the clock to make sure that your quality of life is guaranteed. There are people working in the municipality, they are working in the hospitals, they are working everywhere. Is the water we drink safe? Is the air that we breathe safe? Is the soil where we cultivate safe? Are the milk and beverages that we drink safe? Are the vegetable and fruits that we eat safe? are the chicken, meat, seafood, etc. safe to eat? Are the waste we produce and the industries produce are safely handled, treated, and disposed? These are the main concerns, and I'll be touching upon some of these points as I move on. Each of these things are touched upon in today's talk, and I will also say what are the type of instruments being used for this. Then you will realize the importance of analytical instrumentation. And this is to bring you all consciously. It is not that all of you may be knowing all these things, but it is to make you to have a conscious effort to make sure that we all share the same viewpoint. And you may be able to add on to this, but I don't think you'll be able to delete anything out of this. You are welcome to add on it, and if I can hear your comments, I'll be very happy to even to modify the slides if necessary. Now, what are these compounds really? Now, I am basically here going to concentrate today later on the organic chemicals, but I have mentioned a lot about the metals also today and the instruments that is used for metal analysis. Now, these are organic compounds generally containing carbon, hydrogen, oxygen, halogens, etc., and phosphorus, etc., and inorganic compounds containing alkali metals such as sodium, potassium, calcium, heavy metals such as iron, copper, lead, mercury, and arsenic, and organometallic compounds. These are the compounds we are trying all the time to see whether they are present, whether they have any adverse effect on our health, and so we want to make sure we identify them and we purify so that everything is fine. Now, what are the chromatographic techniques? The chromatography techniques is the very simple one, column chromatography. That's where the chromatography started as early as in 1903. Then we have a very early adopted thin layer chromatography. Then we have early adopted paper chromatography. Then later on we migrated to gas chromatography, liquid chromatography, supercritical fluid chromatography, capillary zone electrophoresis, ion chromatography, etc. Now they have all different applications. Some of them are into small molecules, some of them are into large molecules. Ion chromatography is for cations and anions. So there are many, depending upon your application, we have to use the right technique. Now this many of you may be very familiar with the electromagnetic spectrum. If not, if you are interested in spectroscopy, then it is very important that you download this from the internet and you have it. This electromagnetic spectrum, you can see that we have taken from the lowest, the picometer length all into kilometers, and that is the wavelength. And then these are the size of the molecule, like water molecule here and all that. And these are the rays, these are the energies here. Now these are the gamma rays, then you have the X-rays. So here you will see that here is the gamma rays, and then here is the X-rays, then soft X-rays, then here you have the ultraviolet, and this is the infrared region, and then you have the visible region here in between, and then you have the microwave region, radio waves. These are the things there is less energy, but here is the more energy. This affects X-rays and gamma rays, etc. are harmful to us. And if you see that here are the radio waves, this is where our communication come, they do not affect us very much. And as we move on, the same thing if you see here, now you will see that you have the ultraviolet region, and here is the visible region, right from violet to red. Then you have the infrared, some of the infrared regions here, and then you have the near infrared and mid infrared here and far infrared. We have covered almost all these things in our various talks today. In the chromatography, we have covered all the topics except SFC. We will try to see whether I can get a speaker on SFC. Otherwise, all the techniques are covered. And here also in spectroscopy, all these things have been covered. In the radio frequency, you have the NMR, and we have the NIR as well as Raman here, and visible, UV, FTIR, all these topics we have really covered. And then you see we have talked about electronic excitation, vibration, rotation, nuclear magnetism, and that is the NMR. All these things we have already discussed in the previous webinars. So what are the spectroscopic techniques really? Now if you take atomic absorption, this is mainly for metals, and then you have inductively coupled plasma, that is, and optical emission, icp-oes, and also you have icp-ms, which is very, very important. And then you have the ultraviolet and visible spectroscopy. Then you have Fourier transform, infrared spectroscopy. Then you have nuclear magnetic resonance and mass spectrometry. And then hyphenated techniques such as GCFTIR, GCMS, GCMSMS, LCNMR, LCMS, LCMSMS, icp-ms, icp-ms, ICMS, that is inductively ion chromatography coupled to the mass spectrometer, and icp-ms. So all these type of things are important. We have covered all these topics to you this time. And on the spectroscopic techniques, we have covered everything. And those who have missed our talks, as I mentioned, it will be uploaded onto the website, onto our YouTube channel, and you will be able to see that. This is a lab, how a typical lab, a small laboratory can be built up. And there's an example of the SIS, ICS. I suggest that I recommend that all the universities, all the colleges, if they can afford, they must have a laboratory there, so that it is very important that we teach our undergraduate as well as graduate students, postgraduate students on all our techniques. So what you see here in this laboratory, I will have a close-up of that. And these are HPLCs, GCMS, and UV visible and FTIR. I will focus on some of these things here. Now here what you see is here the GC, and here is the GCMS with the headspace analysis. I will touch upon what is headspace later on in my talk. And then you can see here the HPLCs, all quaternary systems HPLCs, very advanced HPLCs. And then you will see that here is an FTIR, and then there is a UV visible spectrometer, and this is an ion chromatograph. So these are some of the instruments are bought, some of the instruments are even donated to this center. And with that, we are able to help hundreds of teachers and hundreds of students and chemists have been trained in this center for the last many years. So it is a matter of pride that we can say. Now this is to give an idea about this atomic absorption. These are for metals. Now this is the atomic absorption, and this is a flame. And here you can see this is the icp-oes. And I don't think there is a picture of icp-ms right now in this set. This is the high resolution NMR, and you can see the superconducting magnet and the console and the monitor here. They can go up to around 900 to 1000 megahertz now. This NMR has made it very simple, and it is extremely important for the structural elucidations today. When you come into mass spectrometry, LCMS, it is very important that you have the triple quad mass spec. It's a low resolution, but triple quad with a lot of capabilities. And now the latest mass spectrometer is the Orbitrap high resolution mass spectrometer. This has become a very, very, very important instrument today. Many laboratories in India are using this Orbitrap. We introduced this Orbitrap somewhere in 2007 into Bangalore, and today there are many Orbitrap instruments that are available. And I am very happy that during my journey that I've been able to introduce many, many leading, very, very high-end instruments introduced into India, because introducing these instruments to India is not easy, because they have to be supported. So even the manufacturers are a little bit careful to make sure that this can be introduced into India only if they can be supported very well. So we are happy that all the advanced instruments today are available in India. It all started, the chromatography started. The inventor of chromatography is Michael Sweat, who was born in Poland, but became a part of Russia, the Russian scientist. And he is the one who first time used a column chromatography to separate some of the plant pigments, and he called it chromatography, because chroma stands for color and graphy for light. And it was a pure, simple column that he used. And Russian botanist, as I have mentioned here, he lived from 1870 to 1919. He did not get the Nobel Prize. He well deserved it, because Nobel Prize is given only to people who are living. And in 1919 he died. That is the year he was recommended for Nobel Prize. So he died without getting Nobel Prize, but he deserves much more than the Nobel Prize. He changed the way we look at separation science. So this is what he used on the left side. If you see this column, this is the type of things that he used a simple chalk, and he loaded the plant material extract onto that, and he used a petroleum ether to separate them, and they formed a band of colors, chlorophyll and many other plant pigments, and that is separated one by one. So that is basically a simple chromatography, column chromatography, where you have a stationary phase and then you have a mobile phase, which is a liquid. And then you have the TLC, very common again. It was a very early technique where again two-dimensional chromatography, where you have a plate which is coated with an adsorbent, and then you load your materials on a spots, and then you run with a solvent system, and your spots get separated into various components, and then you can see the nature of your mixture. Then you have the paper chromatography on your right side, and that is again useful to many things such as sugars, and amino acids, etc. Some of the things that cannot be used with some of the other chromatography techniques, I think paper chromatography will be very good. And some of those techniques which are maybe used only by chromatography, we are able to do that with liquid chromatography today also. Then we have the gas liquid chromatography, which is abbreviated as GC, where mainly now the predominant GC that we are using is where a stationary phase is a liquid stationary phase and the mobile is a gas. So it is a gas liquid chromatography, which we abbreviate into GC. And then you have the HPLC. HPLC also is basically which is a liquid stationary phase and a liquid mobile phase, and that is a liquid chromatography, and that is what we abbreviated into liquid chromatography. So gas liquid chromatography is abbreviated into GC, and liquid chromatography is abbreviated into LC. Now, what is this important thing in a chromatographic separation? The chromatographic separation is that what is chromatographic separation? In chromatographic separation, two important things are there. One is that you need a stationary phase. Now, stationary phase and then a mobile phase. The stationary phase can be a solid or a liquid, and the mobile phase can be either a solid or a liquid. And by when the mobile phase passes through the solid, then it can separate the components into different individual components. Here is a mixture of two components, 1 and 2, one represented by a triangle, another by a square. Now, this works on two principles. One is adsorption chromatography and another is partition chromatography. Adsorption chromatography is used in TLC, if you remember. There is a solid stationary phase and there is a mobile liquid phase, and that is adsorption chromatography. When you call a GC, where the mobile phase is when a stationary phase is a liquid, mobile phase is a gas, it is gas liquid chromatography, it is a partition chromatography. So also in liquid chromatography, when the stationary phase is a liquid, mobile phase is also a liquid, it is liquid chromatography, it works on a partition chromatography. But if you take a normal column which Sweat used, then he used a calcium carbonate as a stationary phase. So it did not have a liquid stationary phase, it is a solid stationary phase and a mobile liquid phase that works on adsorption principle. So it is very important for one to understand working on adsorption principle and on partition principle. When you take paper chromatography, it works on a partition principle because the water molecule present in the paper are stationary phase, and then you have the mobile phase, which is your organic solvent, and so it is a liquid partition chromatography, and that is paper. Many people get confused with the paper chromatography principle, so I think that is very important for one to understand. Now, when you come to GCMS and LCMS, it's very important to slide that I am explaining to you here. Now, if you are going to plot on x-axis, polarity from low polarity to high polarity, and then you go here onto the molecular weight, you will find that a GCMS can be applied only to volatile substances and that are stable, thermally stable, not labile. So importance to use for GC and GCMS is that your compound should be volatile and compound should be stable, then you can use the GC and GCMS. And if the other things, if it is a non-volatile, some of them can be converted into a derivative. And if you can, for example, if you are an alcohol, you can convert into a TMS derivative, or an acid to a methyl ester, or a alcohol into an acetate, and they or into a methyl ester. And then what happens is it becomes volatile. Then you can use it by GC or GCMS. For example, fatty acid. There were all the partition GC started with the fatty acids and their esters being analyzed as early as in 1948 and 1952. The Martin and Sinch got the Nobel Prize for that, the partition gas chromatography. And then if you see if the molecular weight is high and then the polarity is high, you will find that they cannot be done by GC, GCMS. Then you require HPLC and LCMS. That is why there is a large number of compounds out there which are amenable only to LC and LCMS. That's why those two areas have become very important. So the biggest market today compared to GC and GCMS is HPLC and LCMS. So you will find lots of money being spent on the LC and LCMS because there are large number of compounds, and especially if you are taking biomolecules such as proteins and peptides, they can be done only by HPLC. And these techniques, what I am calling, there are many techniques in LCMS, but now the most important techniques that is being used is the atmospheric pressure ionization. The variation of that is called electrospray ionization. So electrospheric ionization, ESI, and then atmospheric pressure chemical ionization, APCI, and this APPI is atmospheric photoionization. So with this, I move on to the next technique, and that is the basic GC system. So if you have a mixture, and if you have to separate them, then you will have an oven where the oven heats it up, and the injector, you will inject through that, and then the capillary GC column here, which can be about 25 to 30 meter or more, and they get separated here. You heat the column, and then it gets separated, it goes to the detector, and you will be able to see the signal. A very, very, very advanced system, and this is very important to have for volatile compounds. The GC is really a blessing. So again, it is a separation science. Chromatography is a separation science, and spectroscopy is more of an identification science. And if you combine both, that is the best of the worlds. And this is the HPLC both flow path. Then you will find that this is a mobile phase. This is just a cross section. You will have a degasser because many of the solvents has to be degassed. Then you have a pump. Then you have an injector, and then you have a column oven, and then you have the detector. And it is an exploded diagram of the same, and you can use up to different types of solvents. That is why HPLC becomes very versatile, because the selection of gases are very few in GC, whereas n number of solvents can be used in HPLC, including the buffers, organic buffers and inorganic buffers. So it becomes an extremely versatile system, the HPLC. In GCMS, then you have a GC as an introduction point, as an inlet, and then you have the ion source, analyzer, and detector. These things are discussed very much into the GCMS lecture. We talk more about GC in the GC lecture, GCMS and the GCMS lecture, LC in the LC lecture. So we have a separated, dedicated lectures for all this by experts, and I think that is what you will find. If you have missed those talks, you will be able to see them in our videos. And in the liquid chromatography in today's LCMS, the important thing is that you will find that the ionization takes place outside the high vacuum system, and the ions are injected into the mass analyzer and then into the detector. And that is why it is called atmospheric pressure ionization, ESI, electrostatic ionization, which is a variation of atmospheric pressure ionization. So the ionization takes place outside the high vacuum system, and the ions are injected to the mass spectrometer. Very, very exciting indeed. Just to give you an idea, if you have taken a mixture of very common drugs, which is phenacetin and paracetamol, they can be separated by GC, and you will find very good separations here. So in a mixture of two compounds, and you can, if you have standard compounds of this, you can identify the retention time, what is for phenacetin and what is for paracetamol. When you come to HPLC, this is the reverse phase chromatography that we have done. So if it's a normal phase, you will get phenacetin first, and then you will get paracetamol. But depending upon their polarity, this is a reverse phase. So the more polar one separates first, and phenacetin comes second. Very, very interesting. So this is a reverse phase HPLC. It's important to know all these things once you are working with the chromatography. And this is the FTIR. Now we had a very good lecture for FTIR also. So if it is a paracetamol, you can find out, some of the hydroxyl are diffused here, and it works on the functional groups. So you can see NH, OH somewhere here, and then carbonyl around here, 1700, 1750, around 16 onwards. And the advantage is that the fingerprint region, that is a very, very important qualitative technique. So FTIR and mass spectra are the two important qualitative techniques in any quarter flow, because they have what we call as the fingerprint region. When it comes to mass spec, I will explain that also. NMR spectrum, this is the proton NMR spectrum. I don't have the carbon-13 NMR spectrum here. You can take carbon-13 spectrum where it will show all the carbons. Here you will see the protons, and you will see, depending upon the CH3s and depending upon where the H is present and the couplings, you will find either singlet, doublet, triplet, quartet, or multiplet. And similarly, you get the phenacetin NMR spectrum. The idea is just to show this. I'm not going to interpret this spectrum right now, because the NMR spectrum, we had a very good seminar on NMR much earlier. If you take mass spectrum, now this is paracetamol, molecular 151, and that is a molecular ion, and these are the fragment ions. And then this is very unique to paracetamol, just like an IR spectrum. This mass spectrum is very unique to the paracetamol, And then you can compare all this peak to peak. So if it is paracetamol must have this mass spectrum. Similarly, phenacetin, you will find the mass spectra are different. 179 is the molecular weight, and the importance of the peaks start from the right to left. Molecular ion is extremely important than these fragment ions. So this is how you read the mass spectrum from right to left. And these spectra which are lower, around 30s and 40s, they are not very important peaks. All this we will even ignore sometime, but of course you have to see what is a base peak. So it is important to see because this is a 100% relative intensity 100%, and that we call it as base peak, and this have relative intensities based on the base peak. Anyway, these are also explained very well in our mass spectrometry talk. A little complicated spectrum. Now don't think all the mass spectra are so simple. So I have shown this is a cocaine, a little complicated spectrum. You can see 303 is the molecular weight, and then 182, 182. Cocaine, whether you take it in India or any part of the world, you will find the mass spectrum remains the same. And that is like a fingerprint of the cocaine. So that is the importance of that. We can do a library search and we can say that, oh, this is cocaine. And you can interpret the mass spectrum also, the fragmentation patterns, and you can identify what are different ions due to. Yes, I took reserpine. Later on, I'll be talking a little bit about reserpine. Now, this is the LCMS, this is the APCI spectrum of reserpine. This is the electrospray ionization, APCI, and you can see it gives you a protonated molecule, molecular ion +1. And that is what generally it does in LCMS. Again, different types of ionizations are there, either a negative ion or a positive ion. These things are explained in our LCMS talk in detail. Now I will go into a little bit of applications. I'll try to cover as much as possible. If I can cover in 1 hour or a little bit more than that, I will try my best. So this is something that I have invented, which is I call it as a chromatography axle. And what I did was I put around here wheels, and each wheel represents an industry. So if you are working on a particular field and if you are using chromatography, feel free to extend this axle with the different wheels. So here I have mentioned here food and beverages, essential oils, pharma, petroleum, petrochemicals, environmental. You can have synthetic plastics, polymers. You can keep on adding these things, organometallic compounds, whatever you want, you can add, medical sciences. But the chromatography axle remains the same. Chromatography helps in really the separation of your mixture. Now, petroleum and petrochemicals applications, and one is petroleum exploration. The GC starts mainly GC is used in petroleum sciences, and you will find that the petroleum exploration analysis of the crude oil. You want to see how good the crude oil is. What is the amount of sulfur present in that? So that will determine the cost of the crude oil. And the refining of the crude oil, at every stage you need to use the GC. GC is the most used instrument, GC and GCMS, in any of the refineries. Then refinery gas analyzer, hydrocarbon analysis, natural gas analysis, transformer gas analysis is very important in our power stations and all that, or in the power transmissions. You will find that the big transformers are there is some oils, and this oil starts degenerating, and then it forms a gas which is highly explosive. And that is why sometimes when you say that the generators have exploded, and these are some of the gases. So there are some of the GCs, the mobile GCs or small GCs, which are mounted onto some of these big generators, transformers, to analyze continuously online analysis to see whether it is forming any of these gases which will explode. Then the biogas, and then anything, whether it is synthetic plastics, their identification, everything, you need chromatographic methods, mainly GC. Then the environmental applications, when you see the environmental applications, you will find that air monitoring is very important. Ambient air, atmospheric air, and pollution from automobiles, industries, incinerations, everywhere we are polluting the atmosphere. So we must see that there are really right now, there are many monitors available in various highways and many roads within the city to constantly read not only the NO2 or CO2, etc., but all the other organic chemicals which are coming. These are very important to measure that. And then drinking water. Drinking water has got volatile impurities, semi-volatile impurities, and pesticides and river water, lake water, etc. It is one of the most polluted water, whether it is river water, if you think, it is one of the most polluted water. And I think we are doing our best to pollute, but we are also doing our best to make sure that we purify them. And I think there is a lot of rainwater, lot of this dirt coming into even human waste, coming into the river, and that is a very, very sad thing. Wastewater, same thing, drainage water, industrial effluents, etc. I think a lot of we have to be very conscious about it, and wherever the atmosphere that we don't want to put all this drainage water, etc., into any of the river systems or canals. There was a time when I used to go around in the country. I used to see some of the waters in different colors because there will be a chemical industry next to that, and they will put all their effluents into that water, into that river or into that canal, and it will be all colored. Those things have started disappearing now. And again, solid waste. We produce a lot of solid waste and sludges from the industries, and I think we need to worry about that. US EPA has very good methods if you are interested in whether it is air analysis, water analysis, drinking water, wastewater, solid waste, etc. Excellent methods are available from EPA, Environmental Protection Agency in the United States, and I think you should be able to refer to those methods. Now, the development and quality control of agrochemicals, applications in agriculture and livestock. This is very important. This slide is very important because I am going to touch some very sensitive points here. We must be very conscious what we are doing. Agrochemicals is very important. We just use these chemicals very indiscriminately. If they say 1 gram to be put in 10 liters, we will put 10 grams in 1 liter. And that is an abusive use of our agrochemicals into the soil. They all end up in the soil, or they will end up in the vegetables. And so also the pesticides. Pesticides and herbicides, as you know, have ended up, have spoiled many lives in India. Many children are born with the deficiencies, with the problems. And so we have to be very careful. And if they get into the vegetables and fruits, we have to make sure that we are not subjected to, we are not victims of that. Now, one of my friends suggested to me the best way is to make sure the vegetables and fruits are, the vegetables we remove the skin and the fruits we peel off. So for example, things like an orange or a banana would be much safer than eating something which is the skin is also being eaten, like grapes, for example. There are pesticides on the grapes. It's very difficult to remove them. So we have to be very careful. That is where we have to be very careful about how we deal with our life. And pesticide residue and colors in food and beverages, pesticides in Coca-Cola, that was a big problem many years ago. And lead in Maggi noodles, that you are aware of all those things. And the lead things being used in the toys, and the children will put those toys in their mouth and they will get lead poisoning. So the environment, we have to be very, very careful. And commodity inspection such as adulteration of spices such as chili. There was a one time they used to the synthetic red, the Congo red they used to use. Imagine turmeric powder being mixed with the nitroanilins, or you know the color of the nitroanilins. And then milk products, triazines. That was a big problem. Triazine is a 6-member ring with a 3 nitrogen present in that. And these were used as additives in China a few years back. And they executed all those people who are involved, 3 or 4 people who mixed triazines with milk, because they look at the nitrogen content. The quality of milk is seen with the nitrogen content. So, and then oils and vegetable fats, for example, mineral oil being mixed with an organic oil. That is another problem, or animal fat. And many problems, they don't like some of the animal fats being mixed with vegetable fats. But, how do we find out? There are methods to know that. You have to analyze the fatty acids, etc. And when you store the nuts and grains, we have to be very careful. There are fungus which is produced around these nuts. Aflatoxin is one of them, because if you have peanuts which are stored in a wet condition and the fungus grows on that, aflatoxin is one of the most dangerous poisons, toxins. As you can imagine, a few years ago, some of the school students in Bihar died, and that was from aflatoxins. And aflatoxin estimation is done by HPLC. All these things, whatever these things that you are seeing, lead, if you are seeing, it will be atomic absorption, ICP, pesticides, and many organic compounds you will use, GC, GCMS, LC, LCMS, etc. And aflatoxins, it is HPLC with fluorescence detector. And presence of antibiotics and nitroforans. See, this is a very, very big problem. Many of the poultries and the animals, they suffer from a lot of diseases, and then they use a lot of indiscriminate use of antibiotics and nitroforans. They are injected to them, and steroids, etc. What happens to them? And they use sometimes steroids so that the chicken becomes very big, because they are anabolic steroids. So once you inject them, they become very big, and the chicken is sold by weight. So the chicken becomes very big. But what has happened is there's a lot of steroids in that. And when we consume all these things, antibiotics and this, the problem is that it affects our body. We get the steroids in from this meat into our body, and the steroid causes a lot of problem, even inducing diabetes, etc. And antibiotics, as you know, our immunity system comes down. So we have to be very, very careful about that. So our both APIDA and MPIDA are working a lot, making sure our meat, poultry, vegetables, fruits, etc. are analyzed, and then only it is released into the market. So the government is working a lot, but then we have to be very careful, people using these things. And the presence of toxic organic metals in fish, and metals in fish, oysters and crabs. If you dump all these things to the ocean as well as into the rivers, what do? They consume these things, consume, and they ended up, metals end up in fish or oysters and crabs. And one of the cases was mercury ending up in some of the oysters in Japan, and that caused a lot of deformalities in the children in Japan. They are still suffering from that. So the toxic metals, coming from this. So my only request to all the people is that please find out what is the source of any of these things that you are getting. Vegetables and fruits generally not much of a problem, but there are only some of the pesticides, and systematically they can come from the soil into the plant also. So if you are taking, if you are greens, then you have to be careful. For example, palak, etc. You have to be careful because they are growing very close to the soil. But if you are taking a mango, the chances of this pesticide coming systematically into the mango will be very less. But in the case of fish, oysters, etc., you have to be extremely careful because all the chemicals and the metals that we dump into the water, into the river, they end up into the sea, and they get all these things, and we have to be extremely careful. That is a note that I want to give all those people who are attending. We should be conscious, and we should be talking to the other people, educate the people. We are the ambassadors of sharing this knowledge with others. If there are students who are attending this seminar, please make sure that you share this with your other student friends. This is science. So it is important that we share this knowledge with others. And be careful, be very aware. When you are selecting any fruits or vegetables or meat or anything, be aware. And one of the problems in storing the chicken and meat is that when you keep them outside, plenty of bacteria grows in that. And similarly, potatoes. The potatoes, when it is boiled and kept outside, that is the best medium for bacteria to grow. So we have to be very, very careful not to eat those things which are exposed to the outside world, very much into the open atmosphere. We have to be very, very careful. So these are the things that we should take care as people who are from science. I say science background really helps. And these are the things I say science touches our life, analytical instrument touches our life. When I say such comments, I think it holds a lot of meaning. Now, when you take to edible oils and fatty alcohols, etc., you can see that quality check edible oils to determine its price, to see the mixing of one type of edible oil with another edible oil. Because what happens is, if there is an expensive edible oil, then they will try to mix it up with some other edible oil which is cheap. So we have to be very careful that we have to analyze the edible oils to see its saponification, to see what the values we are getting. And then the adulteration of edible oils with non-edible oils, mixing of coconut oil with the castor oil, for example, or with the mineral oils, paraffins. These things are extremely dangerous. And we had a situation where the palm oil which came from some other country, we had such a mineral oils being mixed in that. And we pay for the palm oil, whereas mineral oils are very cheap. So that is the problem. Even for 5 kilos, if you add 1 kilo of the cheap, they made a lot of money. And for industrial use, we use a lot of alcohols and fatty acids to make soaps and other things and detergents. And we use a lot of chromatography and spectroscopy instruments there. Now, when we come to essential oils, oleoresins and herbal products, India is one of the richest countries in essential oils. You know that. We are very happy with the flora and fauna of India. We are one of the richest countries in the world, both for plants as well as we have the seas surrounding the 3 parts of our land, and we are extremely rich in both the flora and fauna. And this industry mainly depends, this flavor and fragrances mainly depends on GC and GCMS for quality determinations, as they consider the percentage of the main constituent is very, very important. For example, mint oil. When they will look at what is the percentage of L-menthol in it. It's very, very important to have the percentage, and the quality of the cost of that oil will depend upon what is the quality. And limonene isomers are the main constituents of orange oil and lemon oil. So they will see whether there is any mix up or any synthetic added. Now when the synthetic, when you synthesize it, you will get DL. So you have to see whether they are mixing up with anything with that. And synthetic flavors are very prominent these days, and the industry depends on that. And the synthetic vanillin, for example, they are all done with chromatography and spectroscopy. And as you know, many of these things are monoterpenes and sesquiterpenes. They are all volatile. So it is very good that we can analyze them with GC and GCMS. And Ayurveda and Unani medicines are manufactured use chromatography and establish their quality and consistency. There are methods now. You know, HPTLC has become a very important tool in making sure that they do that HPTLC methods to make sure that they maintain the quality of the products. When you come to drug research, drug discovery, all chromatography and spectroscopy instruments are used. And in bioanalytical, LC-MSMS is used, mainly quadrupole LC-MSMS. And quality control impurity profiling, HPLC, UPLC with PDR required. And heavy metals, ICP, icp-oes, icp-ms. Volatile impurities, which they use in the API method, is mainly headspace with the GCFID. There are methods for that, the UPA methods. And chiral compounds, LC with the chiral columns. Now the chirality is very important. We did a webinar about 2 weeks back about it, and it is very, very 1 week before. And then very important to know the chiral constituent of that which is active, and the other part is not active. It may cause even danger. And antibiotics, insulin, proteins, etc., HPLC and LCMS. These are the value chains. You may not be able to see clearly this, but it raise a complaint. This is called the value chain of pharmaceutical industry. You can download this from the internet. Please look at the pharmaceutical value chain. Those who are interested in establishing yourself in pharmaceutical company, the students, you should know the drug discovery, drug development, manufacturing, distribution, and then every stage. You can see underneath what are the things mentioned. Chromatography and spectroscopy are extremely valuable things. Without that, the pharmaceutical industry cannot function today. So if you enter into a pharmaceutical industry, you will see hundreds and hundreds of HPLCs, GCs, mainly HPLCs and LCMS present there, and other spectroscopic instruments also. So it's very, very important. Forensic science is a very, very important part. Drug trafficking. You know, in the newspapers every day you read that cocaine has been confiscated, heroin has been confiscated, and they are all crores worth. One kilogram of cocaine, I think it is several crores, and heroin is also same, cocaine is also same, very, very expensive. So if you have to know that, then definitely you need GCMS, FTIR, etc. At least 2 very important techniques to make sure that you have 2 qualitative techniques to confirm which is the product, and then legally they require that. And drug trafficking in many places, they get even capital punishment, especially in Singapore. If the constituent is more than 15 grams of any narcotic substances that one carries, and they are really executed there. They have capital punishment. And drug abuse based on national, that is NIDA. NIDA uses a lot, that's an American agency. Drunk alcohol, this is a big problem. And what is allowed is only less than 0.03%. So we have to be very careful. In India, I said safety pattern. There are a lot of people who drink and drive, which is very dangerous. They should not do that. They are causing problem for others' health. Once they drink, they should not drive. Very simple. And document analysis, ink used. There are always there are arguments in the court that this will is written by so many years ago, this will is written with that ink. So to understand which the ink, and depending upon the different periods, different things are present in the ink, you can find out by a TLC, HPTLC, paper chromatography, etc. And suicide or homicide, both the things, overdose of substances, that this is always a news in India. In every channel that you go, there are always cases of suicide and homicide there. And the chemical toxicology and body fluids, or people trying to suicide, they will take a pesticide or they will take a sleeping pill. We need to understand what is the exact thing there. And once we understand that, then only we can do the overdose. Now, for example, even in the case of a snake bite or a scorpion bite, we need to really take that one, find the blood and find out what type of poison it is so that we can treat that. So there is unbelievable the amount of application that we have chromatography and spectroscopy in our real life. Arson is what? Arson, people take the petrol now. Wherever the hartals are going on, you will find that they will throw the bombs, petrol bomb, or they will burn something. They will put the petrol or diesel or something. We need to find out what is the inflammable liquid used there. And similarly, terrorism. In India, we are used to many countries are undergoing a very big problem due to terrorism. And they are all explosives. Explosives are mainly nitrogen containing compound. Now the RDS, I have put the structure here. Can you imagine this compound? This has got more nitrogen than even carbon present in that. That's why I selected this, this one to show you. And the same way, trinitrotoluene if you take. And trinitrotoluene, of course, the Benson ring is there. Here you see that the six-membered ring has 3 nitrogen itself. It's a really type of a trisine type of a structure. So these are the things in forensic science. It is extremely important that we use a lot of chromatographic science. Even in doping, I will come to the doping thing. One, this is forensic science. If you go into sports doping, again we had a seminar on sports doping by Dr. Alka Biotra a few weeks back, and she discussed a threadbare about all these things. In sports doping, people consume all these banned substances, and we need to find out that from the urine as well as from blood whether they have used any of the substances. And horse racing, they give grams and grams of the steroids and other things, diuretics into animals, and animals don't even know that. They inject into them. And finally, the steroids, they may win the horse may win the race, but one day it is going to crash and die. Greyhound racing, chicken fight, all these things use banned substances. They want to make these animals fight. They want to make these animals strong. So inject all these chemicals into them. And what is the intention of banning the the performance enhancing agencies to establish a fair competition and to save the sports people from the side effects of steroids and similar substances? This is the sadness. Everywhere it is not the sports people who consume this, but it is a team manager or somebody in the country. They say, okay, you can consume this, this is very good. This will enhance your ability. And after winning the race, you will see that this person has taken so many steroids, and then the diabetes will come to him, or he will put on weight, he will grow some of the parts very big in nature, and very sad that they will finally end up in a very, very sad health. Life sciences applications, separations of protein, peptide, enzymes, green chemistry research with enzymes, proteins and peptides, they use a lot of LC and LC-MS. And technique used are ion exchange chromatography, affinity chromatography, gel extrusion, etc. Lot of that. This is very important in medical sciences. I've been reading a lot of that. Estimation of glycosylated hemoglobin. Many of us are doing this test, and that is by ion exchange column. And vitamins are generally all estimated by a lot of information are available in the literature, and please read that. Bilirubin in infants, reverse phase HPLC, and presence and identity organic acid level. This is the unfortunately the organic acid abnormalities in infants, and these are some of the malonic acid, propionic acid, etc., and some of the amino acids also. And that is generally done by GCLMCS. This test is compulsory in abroad, but it is not yet become compulsory in India, and this can be corrected. Otherwise, the children end up in infancy. If you don't attend to it, they will become abnormal children, which is very sad. This is something that we can do when they are born. That is called the neonatal test. Application basic research, as you know that we use a lot of it. Whatever you are doing, students are doing their PhD work, teachers are trying their lot of research work, national research centers, space research organization, defense research laboratories, catalysis, polymer, you name it. They all use a lot of chromatography. I will just next few minutes, I want to change into my gear into going to nature. We need to respect our nature. Respect nature gives us everything, and we have no right to abuse the nature. That is the message that we should share with all others, and that is our responsibility. As science students and teachers and chemists, we must make sure that we protect our nature very, very consciously. You can look at that. Now, this is a willow tree. More than 3,000 years back, they used to take the bark of this tree and boil and drink in China to reduce their fever. And then we got there is salicylic acid, one of the main constituents. And you know, today the largest amount of compound is still synthesized is the acetyl salicylic acid, which is aspirin. And many of the formulations contain that. Ephedra sinica, and this extract of tree bark was used in Chinese some 3,000 years ago. Again, you can see that, and we are using it, pseudoephedrine and ephedrine we use in the cough syrups and other things. So this is the structure of that, just to give you an idea how rich our nature is. This is one of the most important topic that I want to talk to you. This is the periwinkle, or we call it Sada Bahar or Sada Puli. This is present all over in your gardens all around, and we ignore it. But this is one of the most important plants, as you can see. The two important alkaloids, vincristin and vinblastine, were isolated from this. And as early as in 1963 by these people from Eli Lilly, and it is being treated for some types of cancer. And you can see vincristin and vinblastine used in cancer treatment. Some of them are used in basically in even leukemia in children. These are the very accepted drugs. And you see one of them is a CHO, and then vinblastine is NCH3. This is a structure, very complicated structure, alkaloids. And earlier they were using these plants and the leaves even into the type 2 diabetes. I think they find some toxic material element, and I think they are not doing very much into type 2 diabetes, or there are plenty of other medicines today available in the type 2 diabetes. But these are also very effective. Very important, you might have heard about it. I don't know, some of you might have seen these plants also. I have seen it while driving in some of the other countries. This is the papyrus opium flowers and pods, and it is from these pods that you get this, you make an incision, the milk comes out, and you take that on the dark substance after some time, and then you use in making morphine. Morphine is still the best kept secret, the best medicine in the ultimate, when the pain is very acute and it cannot be treated with anything else, especially in the cancer cases, then you will use morphine and morphine derivatives. Very important. And if you are seeing what is now the acetylated derivative of this is heroin. So heroin does not occur in nature. When you do the acetylation of these hydroxyl groups, then you get heroin, which is the diacetyl morphine, 369 molecular weight. The heroin can be done by GCMS, but morphine cannot, because morphine is very polar, so you have to use the LCMS for that. This is very important. And indica, it is very much prevalent in India. This is the marijuana plant, and this tetrahydrocannabinol, this is banned in India, but it is being very much available in UAE, in many other countries, because they are going to make it legal, because one of the best treatment for glaucoma, the eye disease, the pressure that affects the vision, the liquid pressure on the eyeball, that is called glaucoma, and till tetrahydrocannabinol, the cannabis is one of the best treatments for that. This is what they call ganja or bang or whatever. This is the thing. This is very important. This is the cocoa plant. In fact, this does not grow in India. This is more in South America, and I was very happy about 2 years back. I was in Peru. I could actually see the plant. I took many pictures of this plant. This is the coca plant, and they take even this leaves of this, dry it, and they make the tea and take, because that is a high altitude. So they say it is that coca tea is very good for health. That is the source for cocaine. So that is the coca leaves. And this is very important. Sarpagandha is available in India also. This is the source of reserpine. I showed you the structure of reserpine there, the picture. In fact, reserpine is one of the compounds that we use in the tuning of LCMS. We inject reserpine to make sure that we get a good spectrum. So if the LCMS does a good reserpine spectrum, we know that the LCMS is working very well. And that is used for blood pressure and psychiatric disorders. It was used in our long time back in our, even by our local doctors in some of the tribes. They used to take this anticoction and give to people who are suffering some mental disorders. So very well documented, all these things. We are very lucky. India is, again, I want to stress, India is an extremely lucky country to have rich flora and fauna. Pacific U, and very important, and this is the Taxol, which is used in the anti-cancer medicine treatment. And I must draw your attention to Mansukh Wani. Monroe Wan and Mansukh Wani in Research Triangle, they were responsible for the structural elucidation of this. Wani is from India, and I have known him through literature to exchange, but I have not met him. Now there is the Indian neem tree, very well known. Azadirachtin is one of the main alkaloids that is coming out of that. And the interesting thing is that you may wonder why the insects don't come and attack neem. And that is mainly because the antifuran nature of the compounds present in neem. There are many compounds. One of them well known is azadirachtin, and it works as an insecticide in many insect and worms. Today you can buy azadirachtin lotion, and then you can spray into into worms and eggs and all that. They will kill all the parasites. This is a Caribbean coral. Coral became very important, and in 1969 the prostaglandins were isolated from the coral. It was known prostaglandin has got a very important effect in our human body, and it is used for gynecological treatment. It was later on synthesized by E.J. Corey and the group in Harvard. So prostaglandin, today you get the synthetic prostaglandins also, but interestingly this Caribbean coral has got a good about 2% of it in the dried corals. That is very interesting. So prostaglandin, which is very important. It is present in our body. As you can see, we synthesize prostaglandin our body, but this is prostaglandin. Now, going into the fungus, this is Penicillium notatum. Alexander Fleming founded in 1929. It's going to be 100 years very soon, in a few years. And penicillin G and a few of the derivatives are still being used because it developed some type of allergy to some people. But there are a lot of modified penicillin structures today available. Amoxicillin and many things are the derivatives of penicillin. Then this is also bacteria, and this is called streptomycin, and streptomycin is obtained from these bacterial colonies, and that was very interesting. All these things are accidental discoveries, but they are very important. I think they discontinued that is 2012. Again, there may be many competing drugs may be available, so some of them are newer and newer things are coming up. That's fine with us, and we will stick to what is very useful to us. This is a very important one. This is a Brazilian pit viper, and the viper, interesting thing is that when a viper bites somebody, the victim, the blood pressure comes down like crazy, and then finally the patient dies. This victim dies. So somebody investigated what is the reason for this, and then they found out that this guy, Sergio Ferreira, found out in 1965 that this contains the type of compounds like catopril, which lowers the blood pressure, hypertension. So they took the derivatives of that. Today, a lot of drugs, even Cardace and many other things that people consume, one of the largest amount of antihypertensive drugs used for blood pressure is derived from this. Catopril, if you can, Ramakrishnan, and many things are available, and these are all the ACE inhibitor, as you can see. So from viper, we have got a very important. So when we kill the snakes, we have to be very careful that we may not be even knowing what all type of compounds are present in these things. Snake venom is very important, as you know. We have done a lot of research on snake venom, and there are a lot of medicine which are derived from snake venoms. This is a Gila monster. Is very interesting. So in 1990 that this was found out, and this is in type 2 diabetes. Some of the people like Eli Lilly and all that had purchased this patent, and this is a exenatide which is called a peptide. This is the peptide, as you can see, and this is used for diabetes too. I don't know still whether it is being used, but they had some problems with that. But initially it was used. It was a multi-billion-dollar business. I think that's what I wanted to say, that I want to finish here with a note to everybody. First of all, I want to thank you all for attending this webinar today, and it is very important that we pay attention. Today there is no branch of science that which does not depend upon analytical instruments. Wherever you go, any doctors or anywhere, they want to say, even if you go, they want to know the amount of metals present in that. In fact, very interesting to know that our body contains more than 60 elements. That is something, a topic that we would like to discuss sometime later, how our body is a great body with containing a lot of chemicals and chemical substances. So we want to take care of our body and the mind, and what is important is that after listening to this whole talk today, you will find that analytical instruments are definitely helping us to provide us a very cleaner environment. We should consciously look at it, we should consciously invest in it, we should consciously learn about it. And healthier, in medical sciences and other things, I told you, pharmaceutical industry and other medical sciences, how much we use the analytical instrumentation. Today, any doctor, if you have to see, they have to depend upon HPLCs. They will say, I want HB1AC, I want bilirubin, I want uric acid, I want the analysis of this, that. Anything that you take, they are all depending very heavily on analytical instrumentation. We have to really know where they are storing the chemicals, organic chemicals, explosives, etc. We should unearth them, and the police should work on it so that the society is very clean, and society is very safe for us to live on. And that would be my last slide.